So that is an example of negative regulation. Now, lest you think that eukaryotes don't use positive and negative regulation, they do. We just don't use operons. Eukaryotes regulate not only what DNA is transcribed, like prokaryotes do, but eukaryotes also regulate gene expression by modifying the transcripts and also modifying how often the transcript or the mRNA is translated into proteins. So let's take a look at this big overview and then we're going to look at another animation. Okay, so you get a signal in, okay, we'll say that the eukaryotic cell senses that there's lactose in the environment. So chromatin is unwound, the DNA is transcribed. Now, as we learned in previous chapters, we have introns in eukaryotic genes. So those need to be removed. As we also learned in previous chapters, you got to put a cap on and you got to put a poly A tail on. Then from there, the mature mRNA is taken out into the cytoplasm. Now, if it doesn't have a poly A tail or a 5' cap, then it gets degraded. In other words, if a signal comes from outside the cell while the mRNA is being transcribed that says, no, just kidding, there's no lactose, we can get rid of that mRNA without putting forth the energy to make the protein. Okay? Now, if that stays on, then we make a polypeptide. Okay? Protein becomes an active protein. There are signals that say, okay, the lactose is gone, let's get rid of the protein, we don't need to be having it hang around processing something that's not there. Okay, so those are the basic stages of eukaryotic gene expression. Now we're going to watch some animations that go into detail on eukaryotic gene expression and how it's regulated.